In this lecture, we're going to talk about the brain, part of the central nervous system, and we're going to be focusing on the major regions and their functions. So recall in the last lecture we talked about neurons and how they communicate with each other through nerve impulses, such as action potentials. So here we see an actual sagittal section of a brain. That is the type of tissue you're looking at. So there are neurons and neuroglial cells in there. So looking at the major regions of the brain, it's often thought of as having four major regions. The cerebrum, which is also referred to as the right and left cerebral hemispheres. The cerebellum, which looks like a piece of cauliflower on the back here. The diencephalon, which is a kind of a hidden part of the brain in this area I'm circling. And it's made up of some divisions that we'll talk about: the thalamus, the hypothalamus, and also above the thalamus, the epithalamus, not written here, but considered part of the diencephalon. And then the fourth major region is the brainstem, which is this part of the brain here. It's the lower inferior-most part of the brain. So cerebrum, cerebellum, diencephalon, and the brainstem. We're going to be focusing on those. Here's the diagram of those regions. But before we get into the main functions of those different areas, there's a couple other topics I wanted to briefly cover. The brain itself develops in the embryo as a tube, a nerve tube. They call it a hollow nerve tube. And it runs along the back, this posterior part of the body, so they call it the dorsal hollow nerve tube. And the tissue becomes the actual nervous tissue of the brain and also your spinal cord. But the hollowness, the lumen or of the tube becomes the ventricles of the brain. And also the spinal cord has a cavity in the middle called the central canal. And so these are the ventricles of the brain, and they're filled with fluid, cerebrospinal fluid. And they have names, of course. And so let's start with the lateral ventricles. I think you can see those best in this view, this anterior view, where there are two lateral ventricles. These are C-shaped cavities, again filled with cerebrospinal fluid, one in each half of the cerebrum, or the one in each cerebral hemisphere. And then that space is continuous through a little opening here into the third ventricle, which is in this area. That is within the diencephalon. So there's a third ventricle, and then it narrows. That part's called the cerebral aqueduct, and then widens into the fourth ventricle, which is down in the brainstem, and then that flows into the central canal of the spinal cord. So those are the ventricles in the central canal of the spinal cord. Here's a transverse section of the brain showing the two lateral ventricles. And notice they are separated by this septum pellucidum, which means clear. Septum is very thin. Look how thin that is. That separates the two lateral ventricles. Okay, and then also I wanted to mention the connective tissue coverings of the brain called the meninges. Going from the superficial deep, they're called the dura mater, arachnoid, and the pia mater. Dura mater, they're all connective tissue, but the outer layer is a denser connective tissue. In fact, dura means tough, like durable. Tough mother, mater is mother in Latin. The arachnoid is not as dense of a connective tissue, and then the pia mater is a much looser connective tissue. Pia mater means gentle mother. So these are the connective tissue meninges. And here is a picture showing the meninges. So you have to look carefully at this diagram. But the dura mater is in green, and then the arachnoid is this purple line that you see. And notice under it there's a space called the subarachnoid space. There's cerebrospinal fluid in there, just like there are in the ventricles, and then the red line is the pia mater. So those are the meninges. And this cerebrospinal fluid out here, it helps protect your brain. It acts like a shock absorber, because it's actually surrounding your brain. Also, one more thing you notice in this picture is along the sagittal plane, the mid-sagittal plane, notice how the dura mater divides into two layers with a space in between. That is what is called the superior sagittal sinus. It's considered a vein. It is a vein, and what's in it is blood. And I'm mentioning it because it's important in the way cerebrospinal fluid circulates. So in this picture, in this cadaver picture, you see the superior sagittal sinus. So you see it does look like a vein there. And you also see the dura mater. See how tough that is on the outside of the brain. On the other side, they took it off so that you can see the brain. Now, this slide has way too much text on it, but I'm going to use it to briefly go over the circulation of cerebrospinal fluid in the ventricles and in the subarachnoid space. So, cerebrospinal fluid is actually made by a structure called the choroid plexus. And there's actually one in each ventricle. The two lateral ventricles, the third ventricle, the fourth ventricle. See, here's one down in the fourth ventricle. And it's basically ependymal cells, which you might remember from when we went over the 6 types of neuroglial cells, and blood capillaries, and some of the plasma in the blood it comes out of the capillaries and becomes the cerebrospinal fluid. So the choroid plexus is where cerebrospinal fluid is formed from the plasma extracellular matrix of your blood. And so it enters the ventricles, and starting from wherever it's made, it's going to move inferiorly. So for example, if it starts in the lateral ventricle, it's going to go into the third ventricle, down the cerebral aqueduct, into the fourth ventricle, on down through the central canal. And at some point along the way, and you don't need to go into detail here, but at some point along the way, the cerebrospinal fluid leaves the fourth ventricle or the central canal, and it enters the subarachnoid space. So there's some apertures. See, this lateral aperture is one place where that can happen, where it can enter the subarachnoid space. And remember, that's outside the brain on the spinal cord, between the dura mater, or between the arachnoid and the pia mater, rather. So from the subarachnoid space, then, the cerebrospinal fluid is moved by ependymal cells superiorly, and then it is moved out of the subarachnoid space, into the superior sagittal sinus, and at that point it's considered back in your bloodstream. So it comes from your blood and it returns to your blood, cerebrospinal fluid, and it circulates through the ventricles and the subarachnoid space. So it has multiple functions, and here are some of them. It's carrying protective chemicals in it, nourishment, glucose, things like that. It's acting as a shock absorber, especially in the subarachnoid space. So while your nervous tissue is vascularized, it has blood vessels to bring a lot of these same substances. It's also providing nourishment, protection, and bringing various chemicals to the neurons. And notice down here, they say it's continuously circulating. Okay, so you're constantly making it new cerebrospinal fluid and draining away old cerebrospinal fluid into that superior sagittal sinus. Okay, here's just a couple interesting cadaver views of the brain. Notice how highly folded it is. We'll talk about that. Here's another view where it's a sagittal section, And you can see the gray and the white matter pretty clearly in this view. Okay, so what we're going to do next is go through the major regions that I mentioned at the beginning, cerebrum, cerebellum, etc., and talk about their main functions. Again, this is a very cursory overview. We could go into a lot more detail, but I'm touching on the basics of the functional different regions of the brain. So starting with the cerebrum, this is the largest part of your brain. It makes up about 83%. You can see in this picture why they talk about the right and left cerebral hemispheres. There's a big crack down the middle, the longitudinal fissure. On this side over here, you can see the pia is still intact and the arachnoid. On the other side, they removed it, and you can see the folds very clearly on that side. This slide shows that the cerebrum, because it's so large, it's conveniently divided up into lobes, and there's 5 of them, and 4 of them will be easy for you if you remember your skull, because they're named after the skull bones that overlie them. So it's the frontal lobe in red, the parietal lobe in blue, the occipital lobe in green, the temporal lobe in pink, and then the 5th one you can't see, it's behind the temporal lobe, but if you pull that away or cut it off, you see the insula, which is the 5th lobe. So that just gives anatomists a way of being a little more specific when talking about the cerebrum. You can talk about which lobe of the cerebrum. Here they've cut off the temporal lobe so you can see the insula behind it. Okay, and then some vocabulary terms. You definitely want to know what a gyrus and a sulcus is. So the gyri, or the gyrus, singular, are these ridges, nervous tissue, and the sulcus, or sulci, are the grooves in between, these grooves down in here. So it's like the hills and the valleys. And as you might guess in anatomy, the different gyri have names, like the central gyrus, the precentral gyrus, and so on. We will not be covering that in this course. The purpose of these gyri and sulci is to allow you to pack a large surface area of nervous tissue inside your skull. So it increases surface area. And if you look at this picture, this is a frontal section of the brain. The main usefulness of this picture is to show you the arrangement of the gray versus the white matter in the brain. So just to review that, this was something we covered in a previous lecture. Gray matter is primarily neuron cell bodies and dendrites, and perhaps unmyelinated, parts of unmyelinated axons. And white matter contains a lot of myelinated axons with the myelin sheath with all that lipid, so it gives it a whiter color. So that doesn't mean there's no unmyelinated axons in the white matter, or no neuroglia, but it's just what the predominant types of cells or parts of cells that you find. So I do expect you to be able to describe the arrangement of the gray and the white matter in the cerebrum. So the gray, let's start with the gray matter. You find it in 2 places. On the very outside, there's a not very thick, just several millimeters thick gray matter on outer gray matter layer called the cerebral cortex. It's called the cortex, cerebral cortex. And then and then under that is the white matter, which you see in here. And then these purple objects that you see, they're actually gray matter as well. So this is the other place you find gray matter, as islands of gray matter within the white matter, and collectively they're called basal nuclei. Basal nuclei. Alright, so the gray matter once again is in 2 places, the outer cerebral cortex and the inner nuclei, or basal nuclei. Notice they have individual names. We're not going into that much anatomical detail. But they do have, those are their individual names. So you've got gray matter on the outside, islands of gray matter on the inside, white matter in between. And the cerebrum is the one we're going to be talking about, the arrangement of these things in. We won't be covering the other parts of the brain, cerebellum and so on, as much, but just keep in mind that they also have nuclei and white matter and gray matter. This is an actual frontal section of a brain. This is not stained or anything, but you can see the difference between the white matter and the gray matter. Okay, now let's talk about the white matter for a moment. The white matter, as I mentioned, is mostly, but not necessarily completely, myelinated axons. And they come in three types. Really, these three types, association, commissure, and projection fiber tracts, are based on where they're going. So first of all, a fiber tract is a bunch of axons traveling together. So it's many axons going. Like if this is an axon, it would be a bunch of them traveling together in a tract, fiber tract. If those fiber tracts are staying on the same side of the brain, say in the right half or the left half, then we call them association tracts. If they're crossing over the other side but staying in the same part of the brain, like going from the right to the left cerebral hemisphere, we would call them commissure or commissural fiber tracts. But if they're traveling to a completely different part of the brain, like from the cerebrum down to the cerebellum or the medulla or maybe even the spinal cord, then they're called projection fiber tracts. So they're made up of axons. It's really just based on where they're going. As you might guess, the projection fiber tracts are going to be the longest because they're traveling far. Association tracts are the shortest. This is a view that shows you the fiber tracts. You see these white lines? Those are fiber tracts revealed by dissolving away some of the gray matter. So you see they look like white lines. Each one of those would have thousands of axons in them. All right, so let's talk about the function of the cerebrum. So the fiber tracts we just covered, they're mainly carrying action potentials from one place to another. So for communication, they're sort of like the highways of the nervous system, the central nervous system. The basal nuclei, we primarily know their function from looking at many case studies of patients who have had lesions or injuries that affect their basal nuclei, or who have Huntington's chorea or Parkinson's. And from those types of patients, we've learned that the basal nuclei play a big role in coordinating skeletal muscle contraction. So does the cerebellum, which we'll mention later, but the basal nuclei does as well. So now the cerebral cortex is that outer few millimeters of the brain. It has many important functions, and some of them are shown here in this map showing the so-called functional regions of the cerebrum. In other words, different gyri in the cerebral cortex have been associated with specific functions. So this is one of the theories about how the brain works, the regional specialization theory, that specific regions have specific functions. And there's also the more holistic theory that many parts of the brain are required, or the whole brain is required for it to function properly. And the actual way the brain works seems to encompass some of both of those. So there do seem to be some functions like memory that require many, many different parts of the brain working together. But you can also at least start understanding the brain by looking at this functional map. And there have been lots of information gathered, again, based on patients with lesions or damage or actual controlled experiments with using electrodes and stimulating the brain. And they've come up with this map that shows where the neurons are that are responsible for different functions. So the big picture here is that your cerebral cortex is the part of your brain that you use to interact with your environment, the outside world. So this is where, for example, you have your primary motor area. That's this gyrus in red here. These are where your neurons are that allow you to voluntarily control your skeletal muscles. So when you tell yourself, I'm going to get up and walk across the room, you are using neurons in here to give you this command of your skeletal muscles, this voluntary control. So they call it the primary motor area or primary motor cortex. I'm not really asking you to memorize what the name of the gyrus is that it's found in, but I do want you to know its function. And a good way to think about the function of it is to think about what would happen if this part of the brain were damaged. So if you damage your, let's say, your right primary motor area, they found that it would affect your ability to consciously control muscles on the opposite side of your body. So the axons at some point cross over to the other side. So damage to the left primary motor area will cause paralysis on the right side. So that's the function of these neurons. And we'll come back to this map, but let me show you. This is a blow-up of that gyrus with a little picture on the side of different parts of the body. This is called a homunculus map. And what it's showing you is where are the motor neurons, where are the interneurons that allow you to command, for example, the muscles in your hand. So those would be in here, in this part of the cortex. And notice that the hand looks huge compared to the rest of the body because we have very fine motor control over our hand, and also muscles in our face. Notice how big the face is. We have very fine control over muscles of facial expression, and we need it for language. Whereas if you look at your foot, for example, notice it's a lot smaller than your hand because we don't have as fine a motor control over our feet as we do our hands. So there's fewer interneurons in here that are involved in controlling foot muscles. So that's a homunculus map. So going back to this picture of the cerebrum, so that's the primary motor area. Let's cover all the motor areas first in the cerebrum. Next to it, there's something called the premotor area. This is where I like to think of it as an area where you store memories about learned motor skills, or sort of muscle memory. So if you know how to play a musical instrument or knit or do ballet or any kind of, or play tennis or anything you do involving muscles, where you have to practice, and it's a matter of practice, and your muscles remember what movements to carry out. That's information is in the premotor area. So if this area is damaged, then you may lose some pre-learned motor skills. Like you might not remember how to play Moonlight Sonata on the piano. If your primary motor cortex is undamaged, then you'll still be able to move your fingers, but you might have lost some learned motor skills. Also, there's a frontal eye field that plays a role in moving your eyeballs around in the socket. So if you're looking up or down or rolling your eyes, so neurons in there help us command those muscles. And then down here, Broca's area, those are involved, or those neurons are involved in our ability to willfully speak. So the muscles involved in speech. And functionally, they think that your Broca's area is only on one side of your brain and one of your two cerebral hemispheres. So those are the motor areas: the primary motor area, premotor, frontal eye field, and Broca's speech area. Okay, and then so consciously controlling skeletal muscles is important for our ability to get by in the world. Also, our somatic senses, those are, remember from a previous lecture, the senses that we use to monitor the outside world, like vision, smell, touch, hearing, and so on. Those sensory areas are also in the cerebral cortex. So let's start with the primary somatosensory area. That's this blue gyrus next to the primary motor cortex. These are where neurons are that allow you to localize sensory information coming from your skin. So a mosquito bites you on your left forearm, you feel it, and you feel the sting or the bite in that area because it's mapped onto this part of your brain. So it allows you to localize sensory information. You don't necessarily have to look at your arm to tell where the mosquito bit you because it's red there. You feel it. You localize that sensory information. So that's the primary somatosensory area. Next to it, there's an association area that allows you, it's something like the premotor area where it's learned, remembered sensory input. So if I have you close your eyes and I brush your skin with a cotton swab versus an ice cube, it's going to be pretty easy for you to tell the difference. You are able to associate those stimuli with learned stimuli from the past. But if this part of your brain is damaged, you'll have trouble doing that. Back to this primary somatosensory area for a moment, I meant to mention that, like the primary motor area, if your right somatosensory area is damaged, you'll have trouble localizing sensory information on the left side of your body. So again, the neurons cross over to the other side. So for our other major somatic senses, we also have regions of the brain devoted to them. So your primary visual area is back here in your occipital lobe. That's where the nerve impulses from your eyes need to reach for you to see. So, you see as much with your brain as with your eyes, because if the nerve impulses don't reach this area, and these neurons in here, these interneurons aren't functional, you won't see anything. And here again, the axons are going to cross over for the most part. So if your right visual cortex is damaged, then it's going to affect the vision in your left eye, and vice versa. We also have a visual association area that allows us to identify visual objects that we see. So if I hold up an apple, you see it with your primary visual area, but you identify it as an apple using your visual association area. You've seen apples before. So it's against like a memory bank for visual information. The same with the auditory area. There's a primary auditory area where the nerve impulse from your ear needs to go. Damage to your right primary auditory area will affect hearing in your left ear. There's an association area near it where we have auditory memories. So if you hear a train whistle, you can identify that as a train and versus a dog barking, etc. Or the voice of a relative. And then there's also a gustatory area for taste and a gustatory association area. And one that you don't see here because it's in the insula region behind the temporal lobe is the olfactory for smell. So all of our major senses, our the impulses, the nerve impulses reach the cortex. This is where we become, you could say, aware of them. This is where we see and hear and taste and smell. Okay, and then I also wanted to mention two other regions. The common integrative area is this region in here. This primarily, and I'm oversimplifying things, but this primarily integrates sensory information coming from the different regions, like touch, sound, vision, taste, and so on. So it allows us to integrate sensory information so that we have a coherent picture from moment to moment of where we are and what's going on. So damage to that part of the brain can be quite debilitating, because it's not enough just to see or hear or smell. You have to be able to put those together and form, a coherent picture of the environment that you're in at that moment. And then I also wanted to mention the frontal lobe. This area of the brain is associated with personality, behavior, motivation, cognition, sense of right and wrong, our ability to get along with other members of our species. And again, a lot of what we know about this comes from those with damage to this part of their brain, or a stroke, or a lesion, a tumor, or something like that, an injury, and often they undergo pretty drastic personality changes, and usually not for the better. They might become withdrawn or agitated, or go from being mild-mannered to aggressive, things like that. So that's what the frontal lobe is functionally associated with. Okay, so when you look at the big picture here, you see that, again, your cerebral cortex allows us to interact with our environment, both to move through it by commanding our muscles and to perceive it through smell and taste and vision and so on. I showed you that before. This I didn't show you yet, but this is another homunculus map with the primary somatosensory area. So again, notice that our face and our hands are well represented more than our trunk or our foot. Okay, so that's the cerebrum with its cerebral cortex, the basal nuclei, and the white matter. So next we're going to talk about the diencephalon, which is divided into the thalamus, which is shown here, the hypothalamus, which is below it, and you can't really see it because of this marker, this pointer going through it, but that's where the hypothalamus is. And then above it is the epithalamus. So it goes epithalamus, thalamus, and hypothalamus. It makes up your diencephalon. So I'm going to give you the very minimum major function of these different regions. So once again, please do read your book, because this is not meant to replace reading this in your textbook. So one of the main functions of the thalamus is it's the sometimes called the sensory gateway to the cerebrum. In other words, most of your somatic sensory information, like vision and taste and somatosensory information, goes through the thalamus. So it's kind of the gateway, and then from the thalamus it's directed to the right part of the cortex. So the thalamus would direct visual information to your visual cortex and auditory information to your auditory cortex. Smell is the exception. Smell does not go through the thalamus. So gateway for most sensory information going to the cerebral cortex. The hypothalamus, this region down here, has many functions. So I'm just going to list some of the main ones. First of all, I wish you could see it better, but there is a little gland hanging off the hypothalamus called your pituitary gland. And while technically that's endocrine system, not nervous system, it's a good thing to notice it's there, because this is one of the main functions of your hypothalamus, is to control your pituitary gland. We won't go into how yet. That's something you'll cover in Biology 213. But it helps control the pituitary gland, so it has that endocrine function. It also makes two hormones. So while most hormones are made by epithelial cells, there are some neurons that make hormones. We call them neuroendocrine cells. They're neurons, but they have an endocrine function. They make hormones. So oxytocin and ADH are the names. Don't worry about what they do right now. But those are the two hormones made here. So there's really two kinds of endocrine functions the hypothalamus has: making hormones and controlling the pituitary gland. But that's not all it does. It also helps regulate your internal body temperature. So it's like your body's thermostat. It has a thermoregulatory role. It controls various cycles like hunger-thirst, sleep-wake cycles. It's also part of something called your limbic system, which we'll talk a little bit more about shortly here, but that's your emotional brain, as they call it. So it helps mediate emotional responses. It's also has some what they call autonomic control. Now, if you remember, autonomic neurons, we talked about these in an earlier lecture. Those are the ones that control your smooth muscle, cardiac muscle, and also your autonomic sensory neurons monitor your internal environment. So autonomic control means things like controlling heart rate or breathing rate or blood pressure, the internal environment in the body. So the hypothalamus is not your main autonomic control center. That's in the brainstem, but it does have some role there. It does play a role. So those are some of the many functions of your hypothalamus. Okay, so the pineal gland, which is technically considered to be part of your epithalamus, here it is, this little piece of tissue here, and it actually kind of extends up around like that. Your pineal gland is also a neuroendocrine structure in that it makes melatonin, the hormone melatonin. So you'll study that hormone in 213 along with the endocrine system. For now, I'll just say that the pineal gland, it is a neuroendocrine structure. It makes the hormone. They call it the third eye because it receives information about day length. It receives information from your visual cortex about night and day, and it uses that information to regulate melatonin synthesis, like how much melatonin to make. And in humans, melatonin is thought to help regulate sleep-wake cycles. So you'll learn more about that later. So that's the pineal gland, which is part of the epithalamus. So we just went over the whole diencephalon, the pineal gland, the thalamus, and the many functions of the hypothalamus. Okay, and then that takes us to the brainstem, which is divided into the midbrain, the pons, which is here, and the lowest part, the medulla. So you got the midbrain in blue, the pons and the medulla make up your brainstem. For the midbrain, the basic functions are the following. If you look closely, notice there's two bumps in this dorsal part of the midbrain. See those two bumps? They're called colliculi. The upper one is called the superior colliculus, and the lower one is called the inferior colliculus. And briefly, the superior colliculus is important for something they call visual tracking, coordinating head and eye movements. If you're following a moving object, like watching a bee fly around the room, you got to move your head, to keep track of it. That's visual tracking. So damage to this part of your ear would affect that. The inferior colliculus is involved in what they call the startle reflex, where, say, somebody sneaks up behind you and goes boo and you jump. That's the startle reflex. So damage to this part of the brain would affect that. It has a survival advantage to alerting us to unusual, unexpected, or loud noises. So those are the two colliculi. You might see them in your book referred to as the corpora quadrigemina. Corpora means bodies, quad means four. So that's the fancy name for them collectively, the corpora quadrigemina. You're just seeing two here, but remember this is half the brain. So in the other half, there's another superior and another inferior colliculus, hence four bodies. Okay, and then on this side of the midbrain, the ventral side, you have what's called the cerebral peduncle. That's primarily fiber tracts, myelinated fiber tracts. The pons, like the cerebrum, it has nuclei in it. It's gray matter, islands of gray matter within white matter. But I'm going to very briefly summarize the pons by saying that one of its main functions is it has a lot of fiber tracts in it. Pons comes from bridge because it is like a gateway for lots of fiber tracts connecting between like your spinal cord below and the brain above. So they call it the pons. You could definitely go into a lot more detail about what kind of nuclei are in there, but we're keeping it very general here. And then the medulla, the inferiormost part of your brain stem, formerly called the medulla oblongata, but medulla is fine, and so is medulla. People say it both ways. This is an extremely important part of your brain. This is your primary, your major autonomic control center. So this is the main part of your brain that, for the most part, is controlling things like your heart rate and your blood pressure and how many times you breathe in a moment, in a minute. So, major internal functions, processes. So this is your main autonomic control center. So it's your medulla that's, for example, sending out nerve impulses to your diaphragm, telling you to breathe in and then out, say, 18 times in a minute. Your hypothalamus, which remember I said it has some autonomic control capabilities, so it can override the medulla temporarily. So like, let's say you're surprised by something, it might change the way you're breathing. You might go like that instead of your normal breathing pattern. So your hypothalamus would have a role in that. But for the most part, your medulla is calling the shots when it comes to autonomic control. Okay, so that's the brain stem. The cerebellum will be equally brief, so I'm not covering all of its important functions. But one of its main ones is to help coordinate skeletal muscle contraction, which is very complex. If you were to analyze in detail everything that went into your walking across a room, all the different coordinated muscles that were involved, it's quite complex. And so the cerebellum, along with the basal nuclei in your cerebrum, plays a big role in that. So damage to your cerebellum will cause loss of motor control. You might, lose your balance or be wobbly, things like that. Okay, this is an inferior view of the brain. So you can see you're going to be looking at models or slides or models in lab that show this, but here you can see your midbrain in here, your pons, here's the medulla, and then the spinal cord begins from there. So there's the spinal cord. We're going to be covering that in a different lecture, but that is part of your central nervous system. Okay, and then just a few other brief things that I wanted to touch on. So your limbic system is what they call a functional brain system. It's not just one part of your brain like the pons or just the medulla. It involves numerous parts of the brain, and there is some controversy about just what comprises the limbic system, but the main point I want to get across is that it's not just one region. So for example, up here you see how they say it encircles the upper part of the brainstem and part of the cerebrum, some structures on the floor of the diencephalon. So the main point is it's different parts of the brain are involved. And this is your, to put it succinctly, your emotional brain. It mediates the physiological changes that accompany emotions. So, for example, when you're angry, your blood pressure goes up, your breathing pattern changes, your heart rate increases. When you're afraid, your pupils dilate and you start to sweat, or you might your heart will increase, heart rate will increase, and so on. So all these changes with extreme pleasure, you might release certain neurotransmitters like dopamine, and so on. So these are what we think of as emotions, the feelings that go along with pleasure and pain and anger and fear. We'll be talking about especially fear when we get to the fight or flight response later. Okay, so that's your emotional brain, the limbic system. And then the reticular formation is another functional brain system I wanted to briefly mention. Notice it involves neurons in different parts of the brain stem, some in your midbrain, your pons, some in the medulla. And the main functions of your reticular formation are to filter out repetitive, familiar, or weak sensory information headed up to your cerebral cortex. So we filter a lot of it out. It never even reaches our conscious part of our brain, the cortex, where we would be consciously aware of it. And we have to do this. We can't be consciously aware of all the sensory information around us at every moment. The feel of your clothes pressing on your skin, and the sound of the refrigerator in the kitchen, and you have to be able to filter some of this stuff out. So you're going to filter out the familiar, the weak, the repetitive, that kind of thing. So our brain is good at doing that. That's our reticular formation. And then there's also a part of it called the RAS, reticular activating system. That sends nerve impulses to your cerebral cortex when you're awake, and those are shut off when you're sleeping. So part of your reticular formation is involved in maintaining our state of wakefulness, or I suppose you could say consciousness. So that's the reticular formation. So limbic system and reticular formation are these functional brain systems. And you needn't go into too much detail, just basically summarize their functions in a sentence or two. And then the last topic I wanted to touch on are brain waves. So if you look at this picture, you see this patient has electrodes placed on their scalp, on their skin, and it's attached through wires to an electrocardiogram, a machine that can pick up electrical activity coming from the brain. So they also do this from the heart. That would be an EKG or an ECG, electrocardiogram. Recording electrical impulses from the head is an EEG, electroencephalogram. And our body fluids, having electrolytes in them, do conduct this electrical activity from these nerve impulses. So you see here 4 main types of brainwave activity that are commonly recorded, and they call them alpha waves, beta waves, theta waves, and delta waves. And I won't be asking you which have the highest amplitude or the highest frequency. We're not going to go into it in that much detail, but do just look at them. You see how they're different. Like the delta waves are big, they have a high amplitude, but a low frequency. There's not many of them per second, compared to, say, your beta waves. So there's lots of lower amplitude waves in a second. Alpha waves are slower than beta waves, and so on. So they're different. They look different, and they are correlated with certain behavioral or states. So we'll talk about that in a moment. But one more thing I want to say about this first is that This is a kind of a crude measurement in that you're not measuring electrical activity from a given neuron in the brain. This is more of a global, collective brainwave activity. So here are the states that they're associated with. Alpha waves are usually what are recorded when people are resting, relaxed, or maybe right before you're drifting off to sleep. You would normally record alpha wave activity. Beta wave activity is what hopefully you have right now. This is when you're focusing, concentrating, your most awake, focused self. So you have lots of brain activity going on. Theta wave activity is common in children, but in adults is mainly connected with frustration. So I don't really know what that's saying about children and frustrated adults, but that's called theta wave activity. And then delta waves are associated with deep sleep. So the fact that they're high amplitude, low frequency, what this is really telling you is that when you're asleep, there aren't as many different neurons sending out nerve impulses. There are fewer, but they're synchronized, more synchronous, so you get these bigger waves, wave activity. So the importance of this, and I mention this because it can have diagnostic value. If someone's having neurological problems, then they may do an EEG to look at their brainwave activity and see if it looks normal. So that completes this lecture on the brain.